Welcome friends. The topic for discussion in this video is the mechanism of hormone action. I am Dr. Omkar Singh. Hormone is a chemical messenger. Cell-to-cell communication occurs in three steps: reception, signal transduction, and cellular response. The endocrine cell releases the hormone directly into the bloodstream. It circulates and comes into contact with all cells in the body. The target cells respond. The target cells are cells which carry receptors for the hormone. Other cells which do not respond because they do not carry receptors for the hormone. For example, this blue cell carries receptor for the hormone A, so it will respond to hormone A. It does not carry the receptors. It does not carry the receptors for the hormone B, so this cell will not respond to hormone B. Similarly, this orange cell, it has got receptors for the hormone B, so it will respond to hormone B, but it does not carry receptors for the hormone A, so it will not respond to hormone A. This cells on the extreme right, this green cell carries receptors for hormone A as well as receptors for hormone B. So it will recept to either of the two hormones. Receptor is a macromolecule and each target cell carries about 2000 to 1 lakh receptors, and these receptors are highly specific for their ligands. Receptors are classified based on their location as intracellular receptors and cell surface receptors. The first step of hormone action is the binding of hormone to its specific receptor. The receptors for the steroid hormones and the thyroid hormones, they are located inside the cell, either within the cytoplasm or inside the nucleus. Steroid hormones are lipophilic. They enter the cell by simple diffusion across the cell membrane. Thyroid hormones are derivatives of amino acid tyrosine and they enter the cell via facilitated diffusion. Receptors for steroid hormones are located in the cytoplasm. Receptors for thyroid hormones are located in the nucleus. The first step in hormone action is the binding of the hormone with its specific receptor. When the hormone binds to its receptor, it results in a conformational change in the receptor and activates the receptor. The activated intracellular receptor becomes competent to bind the DNA. Hormone response elements are short specific sequences of DNA. Steroid hormone receptors recognize a base sequence AGA, ACA, whereas the thyroid hormone receptors recognize a base sequence AGG, TCA. The activated receptors bind to hormone response elements. Hormone response elements are specific base sequences within the promoters of the hormone responsive genes. In most cases, hormone receptor complexes bind DNA in pairs, resulting in the formation of homodimers or in the formation of heterodimers. The transcription from those genes to which the receptor is bound is affected. Usually the receptor binding stimulates the transcription. However, sometimes receptor binding inhibits the transcription. Therefore, the hormone receptor complex functions as a ligand-dependent transcription factor, either stimulating or repressing gene transcription. For example, this steroid hormone molecule, it diffuses freely, easily through the cell membrane and binds to the receptors located within the cytosol. When the receptor binds to the ligand, it undergoes a conformational change and becomes activated. Activated receptor is competent to bind DNA. The receptor hormone complex then translocates to the nucleus, where it recognizes its response element, that is a specific sequence of bases on the DNA located in the promoter region of the responsive genes, and then it either stimulates or represses the transcription of the genes, resulting in altered rates of transcription and translation of protein molecules, altered rates of transcription and formation of messenger RNA. The other way in which the receptor steroid hormone can alter transcription is by binding to and activating or repressing other transcription factors. That means instead of binding to intracellular receptors, it binds with transcription factors and those transcription factors either get activated or they get repressed. The cell surface receptors for the protein hormones and catecholamines exert their action by binding to cell surface receptors. Protein hormones are polar molecules. They cannot diffuse across the cell membrane. As such, their receptors are located on the cell membrane. Similarly, the catecholamines like epinephrine, norepinephrine, and dopamine are polar molecules. They also cannot diffuse across the cell membrane, so their receptors are located on the cell membrane. All cell surface receptors have got an extracellular domain which binds to its specific ligand. These receptors also have got a transmembrane domain. This transmembrane domain anchors the receptor to the lipid bilayer, and then these cell surface receptors have got an intracellular carboxy terminal domain. This domain is linked with intracellular signaling molecules. The function of the receptor is to recognize and respond to its specific ligand. There are three common types of cell surface receptors. These include the G protein couple receptors, enzyme link receptors, and ion channel link receptors. This is the diagram of a G protein couple receptors. It has got an extracellular amino terminal domain. This domain binds to its specific ligand. There are 7 transmembrane domains in this G protein couple receptors, which anchor the receptor to the cell membrane, to the lipid bilayer. Because this receptor is passing in and passing out of the cell membrane 7 times, that is why this receptor is also known as 7 transmembrane receptors. And because it snakes through the cell membrane multiple times, it is also known as serpentine receptors. The intracellular domain of this receptor is associated with a heterotrimeric G protein. The next most common type of cell surface receptors are the enzyme linked receptors. For example, receptor tyrosine kinases, which bind to the insulin molecule, and then there are cell surface receptors which don't have catalytic activity of their own, but they are linked to intracellular enzymes which function as tyrosine kinases. Third common type of cell surface receptor is the ion channel link receptors. These receptors are most commonly found on the postsynaptic membrane. Now the First step of hormone binding with the cell surface receptor is the first step in hormone action involving the cell surface receptors is the binding of the hormone with the receptor located on the cell membrane. The binding of the receptor with the hormone results in a conformational change in the receptor, and the receptor gets activated and results in a cascade of biochemical reactions that culminates in cellular response. The characteristic feature of this biochemical cascade is that at each step the signal gets amplified so that a very small concentration of the hormone results in a very large cellular response. The hormone receptors are regulated. Down regulation of the receptor results when the target cell is exposed to a sustained high concentration of the hormone. The mechanism of down regulation is either deactivation of the cell surface receptors or or inactivation of the receptors can either be upregulated or they can be downregulated. Downregulation of the receptors result from sustained exposure of the target cells to high concentration of the hormone. The mechanism for downregulation is either inactivation of the cell surface receptors or the inactivation of the downstream intracellular signaling molecules, or the receptor get internalized so that it is no longer in contact with its ligand. There can be increased degradation of these receptors by the lysosomal enzymes, and receptor downregulation can also result from decreased synthesis of the receptor molecules. When the target cell is exposed to a low concentration of hormone, in that case, the receptors can get upregulated, and the mechanism for upregulation of the receptor involves either the increased synthesis of the receptor molecules themselves or increased synthesis of the downstream signaling molecules. In the next video, we'll talk about the signal transduction by G protein coupled receptors.